Welcome to Chapter 5's video lecture overview. What you're seeing right now is a spherical model of the cell membrane that we're going to be talking a lot about today. And when I see this, I sort of think of the Great Wall on the outer layer of Minas Tirith, which is the cell membrane. And here are the marauding hordes of orcs that are trying to get to the soldiers of Gondor. Now, that's quite the analogy, but what we have is a bilayer of lipids that are self-regulating based on chemical properties, how water molecules move in and out of the cell. And a big part of today's understanding is knowing that cells maintain an internal environment. We call this homeostasis, which means that they're going to be regulating the movement of molecules and energy and signals in and out of the cells by using the membrane's chemical nature. More on that in a little bit. So the membrane that I just alluded to, the cell membrane, is made up of phospholipids. And phospholipids are the most abundant lipid in most membranes. We'll talk more about some variations that we see in that. And they're known as ambipathic molecules. Ambipathic molecules means that they are dual-natured in terms of their biochemical properties. They both have a hydrophobic and a hydrophilic region that allows them to not only self-regulate the cell internal environment, but also they can associate with the cytoplasm and the extracellular fluids. This creates a very stable boundary between two aqueous compartments or two aqueous or water-based environments. And we can see that by looking at this diagram of the phospholipid bilayer where the fatty acid tails are pointed inwards, and then the glycerol and phosphate groups point externally outwards to both the inner cytoplasm of the cell and the extracellular fluids, which tend to be water-based. Now we're going to talk about things like cholesterol and peripheral proteins and glycolipids and integral proteins today and how they function within the cell wall or castle wall, so to speak, of a cell. Now, part of the cell membrane is the role of membrane carbohydrates that are used in cell-to-cell recognition. So cells not only produce proteins, but they also create very specific carbohydrates in terms of antenna or receivers that they embed in the membrane. And they can either be bound to lipids, known as glycolipids, or proteins, known as glycoproteins, that point inwards and outwards to receive information. These vary among species, individuals, and even cell types within a species, meaning your liver cell has different glycoproteins than maybe a smooth muscle cell, based on the types of signals that are going to be sent to those two cells and their roles within tissues and organs. Now, we can make the membrane more or less fluid. The reason why we want to have a membrane fluid is that there are a variety of things that can actually diffuse between either aqueous solutions without energy or any special transportation. The problem, though, is that fat as a molecule is fluid based on temperature. If you think about going home and frying an egg and you put butter in the pan, the solid butter becomes liquid as you heat it up. So as temperature increases in a membrane, we tend to see membranes become more fluid, and as they become colder, the membranes become more viscous, and this can be really problematic in terms of diffusion of gases and nutrients in and out of cells. So in order to make cells more fluid, maybe because temperature is going down, we might pack unsaturated fatty acids into the membranes by selectively creating phospholipids to sort of space out the cell. Alternatively, if we're worried about really high temperatures in cells or tissues, we might increase the saturated fatty acids in the phospholipids to help condense it. And we see actually these membrane changes happen in plants during the winter and summer months. We can also embed cholesterol, which helps reduce packing or viscousness in the membranes, and they're very good at sort of hindering solidification in cold temperatures, but also they moderate fluidity at moderate temperatures. They keep it just right in terms of that fluidity. They're sort of like middle school teachers at a middle school dance or a high school dance, so to speak. They make sure that, you don't have too many people congregating all at one time. They keep the mosh pits separated, so to speak. I'm not sure if mosh pits are a thing anymore, but they were at my high school dance. Now, in terms of the proteins that we find within membranes, we have a couple different flavors. Integral proteins, or you could call them transmembrane proteins, penetrate the hydrophobic interior of the lipid bilayer. We find them throughout the entire membrane. They're sort of like a tunnel or a gate. The hydrophobic region of these proteins tend to be stretches of nonpolar amino acids, remember chapter 2 and 3, that allow them to interact with those hydrophobic fatty acid tails, while hydrophilic regions are in the interior and exterior section of those phosphate groups. Peripheral proteins are loosely bound to the surface of the membrane, so they don't go fully into the entire layer, and they tend to be more useful for either cellular communication via internal or external signals. And we can see here that when we look at a transmembrane protein, there will be different folds of proteins that allow them to interact with both the internal and external biochemistry of the membrane. Now, in terms of different proteins that we find in cells, we can have transport proteins that might be useful in bringing things in or out of a cell, maybe solutes or energy. We can have enzymatic membranes that do specific cellular and chemical reactions. We can have proteins that are very useful for being attached to the ECM, the extracellular matrix, and the cytoskeleton, so sort of creating that anchor between what appears to be actin microtubules to collagen and fibronectin. This creates tissue cohesiveness. We can have cell-to-cell recognition, so a glycoprotein can be used for immunity and defense. So when you have a cold, your body creates killer T cells that use these glycoproteins to go around and find specific bacteria with the same presenting antigen. We can have intercellular joining, so this helps create tight junctions between different cells, often seen within things like your epidermis, your skin, or your epithelial cells in your small intestine, preventing leaking. And we can have signal transduction. So we receive messages, maybe an email from another long-lost relative cell in another tissue, and that leads to some response. Now, reminder about chapter 4, we have the endomembrane system that uses the rough ER and transport vesicles and the Golgi apparatus to modify and produce proteins that can either be secreted or embedded within the membrane for those types of proteins that we just discussed. Transport proteins are going to be really useful for anything that is hydrophilic across the membrane. So water is not going to be easily able to enter into cell membranes due to its polar nature. So we need proteins like aquaporins to get water in and out of the cells. It's kind of fascinating that the universal thing that all life needs can't actually get into the cells easily. Diffusion is going to be the thing that we talk about first in terms of the movement of substances in and out of cells. And diffusion is the tendency for molecules to spread out evenly into an available space. You all know what diffusion is if you have smelled a skunk that's been hit by a car, or you've gone by a farm that just has applied manure. The gas particles are diffusing out from a concentrated space to a concentration that is lower, i.e. your car or your lungs. Each molecule moves randomly within that population of molecules, but it is often directional in terms of a high to low concentration. Once we achieve equilibrium, the molecules aren't going to be moving in a net direction. That often doesn't happen when you have things like an open system, like a manure applied to a field in the atmosphere, but in terms of cells, we often see that molecules might achieve equilibrium where there's no gain or loss of molecules in or out of the cell because they're moving across at equal rates. And diffusion can occur by thinking about things moving in or out of a selectively permeable membrane. So if I have two solutes of molecules in two different solvents with water with no dye here and lots of here, it will randomly move until it reaches equilibrium. And the same can be true if we have two or more solutes in terms of reaching the equilibrium of concentration. Now, osmosis is the movement of water. So diffusion is just anything that's moving from a high to low concentration. But osmosis is the diffusion of free water across a selectively permeable membrane. Wow, those are a lot of words to break down. So let's talk about it. When we talk about free water, we're talking about water that is not currently in some form of association with different solvents. Remember, water is a very polar molecule, so it can easily form hydrogen bonds with things like salt or sugar, forming those hydration chains or those hydration sort of circles around different solvents. Water is going to diffuse across a membrane from regions with lower solutes to higher solutes until the solute concentration is equal on both sides. What the heck does that mean? So I have two solutions of water, one that has a lower concentration and one with a higher concentration. And we notice that over time, the water is moving out of this container and going across this container. Two things are happening to make this happen. One is that the solvents, the actual water, is able to pass through the selectively permeable membrane, but the solute can't. So if this is sugar, sugar can't pass this membrane, so we can't have a diffusion or a net equilibrium of sugar molecules between these two concentrations. So if the solutes can't move, then the solvent will as a result. And basically, because we have more free water, hello, look at all these water molecules that need a friend, they're going to move in at a net rate across the membrane to go free up and associate with some of these solutes. So free water is really a notion of how many water molecules don't have hydrogen bonds with other solutes. A low free water potential means that lots of water have lots of solutes. A high free water means that there's a lot of water with very low solutes. So as you are making your different beakers for salt and sugar, the more sugar and salt you put into your beakers, the less free water was available. Now this has some connotations and some implications for how cells work in systems. So if we put cells in a very low solute concentration or concentrations with a lot of free water, water is going to flow into the cell, causing what's known as cell lysing. And we call these hypotonic because the cell swells like a hippo. And that's because water is more freely available in the external solution, so it flows into the cell. The opposite is true with hypertonic. Because there's more free water available in the cell and not the external environment, water is going to leave at a net rate, causing the cell to shrivel. We call this hypertonic because the little e gets smaller. And isotonic is where we have net equilibrium, so water moves in and out at the same rate. Lots of freshwater organisms have to be conscientious of this because the water concentration is more free than the internal cells, meaning that cells have more dissolved solutes than the fresh water around them. So they have to use things like contractile vacuoles or specific types of nitrogenous waste excretion to get rid of that extra water. Now, in facilitated diffusion, we have things that are going to make the process happen faster. We're going to be making the passive movement across the membranes a little bit faster, a little bit easier. And channel proteins are one way that will allow us to have things flow across the membrane in a very specific direction to make things work faster. One might be aquaporons for the facilitated of diffusion of water, meaning that we're going to have these really specific channels that allow a lot of water to flow in or out of the cells. And others might be ion channels that allow for things like sodium or potassium or chlorine to flow in and out of the cells quickly, which we actually use with neurons to have neural firings, meaning stimuli and responses within our cells at a very rapid rate. In terms of channel proteins, we can have proteins that are an open channel, or we can have carrier proteins that selectively open and close, sort of like a castle gate, to put those solutes in or out of the cell. And sometimes we have to invest energy, such as ATP, to pump things in or out of the cell. So we have a sodium-potassium pump, which is very common in many neurons, where we're going to put some sodium in the channel protein and use some ATP to pump it out of the membrane. And then we're going to use some energy to get the potassium into the cell. And this is happening against their concentration gradients. So sodium is low in the cell, and we're using energy to pump it against its membrane potential. And the same thing is true for potassium, hence why we have to use energy. We're going against the gradient. And this is why we call it active transport, because we are not going from a high to low concentration gradient, and then instead we're going from a low to high. We have to invest that energy to make the solutes work against what they normally would like to do. Sometimes I feel like being a teacher is sort of like being like an active transport protein. Ideally, many of the students here would love to not have any homework. It's up to me to invest energy and time and resources to make them do something that they might not normally want to do on their own. Now, we call these things that we've just talked about, the two combined forces, the electrochemical gradient, which is all about driving the diffusion of ions across a membrane. And we can think about this based on a chemical's force, what's the concentration of that gradient, and the electrical force, what does that have on the effect of membrane potential on the ions of movement. So really we're thinking about when we're looking at different charged particles across the membrane, how is that impacting how a cell functions, usually a neuron. And obviously we can use that energy to make things become either negative or positive. So oftentimes we will use energy to pump hydrogen ions out of a cell to create a positive electrochemical gradient. And because we have all of this charged positivity on the external of the cell with a lot of negative charges in the cell, the positives are going to want to flow back in. And we actually use this mechanism to power ATP during cellular respiration. More on that in chapter 7. And this obviously can be used to do work. So if we create an electrochemical gradient by using some energy to pump out a bunch of positive hydrogen ions into the extracellular matrix, notice that they're positive because it's a hydrogen ion with an extra proton. We've removed an electron from these atoms. They're going to want to flow in, and this net flow going from high to low can be used to get sucrose into the cell. And we call these co-transporters, where we have a passive flow of a molecule, hydrogen ions that want to go from a high to low concentration, positive to negative charge, to get something like sucrose, which does not associate well with the phospholipid bilayer, into the intercellular cytoplasm. And obviously cells can do other things besides diffusion, active or passive, to get things in or out of the cell. Oftentimes you can have phagocytosis with larger food particles or even cells hello dendritic white cells in your immune system. They can swallow other cells and break them down with their lysosomes. We can have pinocytosis, where we take in extracellular fluids. And we can have receptor-mediated endocytosis, where we're actually using specific receptors, glycoproteins, to bring in specific molecules when they're found in the environment. I'm going to stop there and make a new video for cell signaling, just because it is also a complicated topic.